Let's continue from the previous session. Brassinosteroids. These hormones are involved in cell elongation and division, differentiation, and stress responses. They also promote flowering, seed development, and root growth. Then jasminoids. These hormones play a role in defense responses against herbivores and pathogens. They also regulate development such as root growth and pollen maturation. Overall, the functions of plant hormones are interconnected and highly regulated, allowing plants to respond and adapt to changing environmental conditions. Overall, the functions of plant hormones are interconnected and highly regulated, allowing plants to respond and adapt to changing environmental conditions. Then, phytochrome blue light response. Phytochromes are photoreceptor proteins that absorb and sense light in the red and far-red regions of the electromagnetic spectrum. They play a critical role in regulating plant growth and development, including germination, photomorphogenesis, shade avoidance, and flowering time. Blue light, on the other hand, is absorbed by cryptochrome photoreceptors and also plays a vital role in plant growth and development. The blue light photoreceptors, cryptochromes, are involved in regulating various aspects of plant growth and development, including seed germination, stem elongation, and stematal opening, and the regulation of the circadian clock. Cryptochromes are also involved in the perception of blue light, which is important for phototropism and the regulation of flowering time. The phytochrome family is divided into two main types, namely phytochrome A and phytochrome B. Phytochrome A, phyto A, is sensitive to far red light. Phytochrome A is sensitive to far red light and plays a role in regulating germination and seedling development. Phytochrome B, phyto B, on the other hand, is sensitive to red light and plays a role in regulating various aspects of plant growth and development, including stem elongation, leaf expansion, flowering time, and regulation of the circadian clock. Upon absorption of light, the phytochrome undergoes a conformational change and is converted into its inactive form PR to its active form PFR. PFR then moves into the nucleus where it interacts with a transcription factor called PIF3, phytochrome interacting factor 3. This interaction results in the activation of or a repression of various target genes, leading to changes in plant growth and development. Blue light, on the other hand, is perceived by the cryptochrome photoreceptor, which undergoes a conformational change upon absorption of blue light. This change allows the cryptochrome to interact with various signaling proteins and initiate a signaling cascade that leads to changes in gene expression and ultimately plant growth and development. In summary, the phytochrome and blue light signaling pathways are important for signaling and regulating various aspects of plant growth and development, including germination, seedling development, and stem elongation, leaf expansion, flowering time, and the regulation of the circadian clock. Understanding these pathways is critical for the development of new technologies to improve crop yields, and increase plant productivity. Then circadian clock. The circadian clock is an internal biological mechanism that regulates the physiological and behavioral processes of living organisms on a 24-hour cycle. It is responsible regulating sleep-wake cycles, promote hormone production, metabolism, and other biological processes. The circadian clock is present in almost all living organisms, including plants, animals, and even bacteria. The circadian clock operates through the series of molecular feedback loops that involve the activity of specific genes and proteins. In plants, the circadian clock is involved in regulating the timing of a variety of processes, including photosynthesis, stematal conductance, and hormone production. There are several key components of the circadian rhythm in plants, including the transcription factors CCA1 and LHY, which activate the expression of other clock genes, including TOC1 and GI. TOC1 and GI in turn regulate the expression of other clock genes, creating a complex network of feedback loops that help to keep the clock ticking. One of the key functions of the circadian clock in plants is to synchronize physiological processes with environmental cues such as light and temperature. This allows plants to anticipate changes in their environment and adjust their physiological processes accordingly. For example, the circadian clock is involved in regulating the opening and closing of stomata, which helps to regulate water loss in response to changes in light and temperature. The circadian clock also plays an important role in regulating plant growth and development. For example, the timing of flowering in many plant species is regulated by the circadian clock. In some species of plants, such as Arabidopsis thaliana, the circadian clock regulates the expression of genes involved in the production of the floral hormone florigen, which promotes flowering. Overall, the circadian clock is a critical component of plant physiology, helps to regulate a wide range of biological processes and allowing plants to adapt to changes in their environment. Understanding the molecular mechanisms that underlie the circadian clock in plants may have important implications for agriculture and plant breeding, as well as for human health, given the importance of circadian rhythms in human physiology. Then, plant circadian clock. The circadian clock in plants is an internal timing mechanism that regulates various physiological and biochemical processes in response to environmental cues, particularly changes in day and night cycles. It is crucial for the survival and growth of plants as it allows them to participate and adapt to daily and seasonal changes in the environment. The plant circadian clock is made up of a complex network of genes, proteins, and small molecules that interact with each other in intricate ways to maintain accurate timekeeping. One of the most important components of the circadian clock is the transcription translation feedback loop, which involves the activation and repression of key clock genes through protein-protein interactions and DNA binding activities. The plant circadian clock plays a critical role in regulating a wide range of physiological processes, including photosynthesis, flowering, leaf management, stomatal conductance, and hormone biosynthesis and signaling. For example, the clock controls the expression of genes involved in the light reactions of photosynthesis, allowing plants to optimize their energy to use in response to changing light conditions. The circadian clock also regulates the timing of flowering, ensuring that plants flower at the optimum time of year to maximize pollination and seed production. One of the most important aspects of the plant circadian clock is its ability to synchronize with environmental cues, particularly light and temperature. Plants use a variety of sensory systems, including phytochromes and cryptochromes, to detect changes in light levels and adjust their circadian rhythms accordingly. Additionally, the clock is sensitive to temperature changes, allowing plants to adjust their growth and metabolism in response to seasonal fluctuations in temperature. In summary, the plant circadian clock is a complex and essential timing mechanism that regulates a wide range of physiological processes in response to environmental cues, particularly changes in light and temperature. Its importance in plant growth, development, and adaptation to changing environmental conditions cannot be overstated. Next, secondary metabolites. Plant secondary metabolites are compounds synthesized by plants that are not essential for basic metabolic functions but play important roles in the plant's interaction with its environment, including defense against herbivores and pathogens, attraction of pollinators and seed dispersers, and competition for resources. They are often synthesized in response to specific environmental cues and stresses such as attack by herbivores or pathogens, exposure to UV radiation, or nutrient deficiencies. There are many different classes of plant secondary metabolites, including alkaloids, flavonoids, terpenoids, phenolics, and glucosinolates, among others. Each class has its own distinct chemical structure and biological activity, and many of these compounds have been shown to have beneficial effects for human health, such as antioxidant, anti-inflammatory, and anti-cancer properties. One important class of plant secondary metabolites is alkaloids, which are nitrogen-containing compounds that are often toxic to herbivores and other animals. Examples of alkaloids include caffeine, nicotine, and morphine. Another important class of plant secondary metabolites is flavonoids, which are pigments that give flowers and fruits their bright colors. Flavonoids have been shown to have a wide range of health benefits, including antioxidant and anti-inflammatory effects, and are found in many commonly consumed foods such as berries, tea, and wine. Plant secondary metabolites are also important in traditional medicine and have been used for thousands of years by different cultures to treat various ailments. For example, the bark of a cinchona tree, which contains the alkaloid quinine, has been used for centuries to treat malaria, and the roots of the yew tree, which contain the alkaloid taxol, are used to treat cancer. Next, plant nutrition. Plant nutrition refers to the study of how plants absorb and utilize nutrients from their environment to maintain their growth and development. Essential nutrients are required for various physiological processes such as photosynthesis, respiration, and protein synthesis, and are classified into two categories like macronutrients and micronutrients. So, macronutrients are required in large quantities and include nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Micronutrients, also called trace elements, are required in small amounts and include iron, manganese, copper, boron, chlorine, and molybdenum. Plants obtain these nutrients through their roots, either from the soil or from other organic matter. The availability and uptake of these nutrients are influenced by several factors, including soil pH, temperature, water availability, and the presence of other elements. The deficiency of or excess of any of these nutrients can lead to various physiological disorders and affect plant growth and yield. For example, nitrogen deficiency can lead to stunted growth and yellowing of leaves, while potassium deficiency can cause the development of weak stems and increased susceptibility to diseases. Plant nutrition is important in agriculture as it plays a crucial role in productivity and quality of crops. Understanding the nutrient requirements of plants and optimizing their uptake is essential for sustainable agricultural practices and ensuring food security. Various strategies are employed to optimize plant nutrition, including the use of fertilizers, crop rotation, cover cropping, and organic farming practices. These strategies aim to maintain soil health, enhance nutrient availability, and reduce nutrient losses, thus ensuring the long-term sustainability of agricultural systems. In conclusion, plant nutrition is a fundamental aspect of plant biology. So, phloem loading. Phloem loading is a process by which photoassimilates like carbohydrates produced in the source tissues during photosynthesis are transported from the mesophyll cells of leaves to the seed tube elements of the phloem for translation to the zinc tissue like growing regions of the plant such as roots, developing fruits or seeds. The process involves the active transport of photoassimilates from the mesophyll cells to the phloem parenchyma cells and then from the phloem parenchyma cells to the seed tube elements. Source sink relationship. Phloem loading is an important process of plants to establish and maintain the source sink relationship between different plant organs. The source organs are the sites of photosynthesis, while the sink organs are the sites of carbohydrate utilization for growth and development. Symplastic and apoplastic pathways. There are two pathways for phloem loading, the symplastic pathway and the apoplastic pathway. The symplastic pathway, photoassimilates move through plasmodesmata, while in the apoplastic pathway, photoassimilates move through the cell walls. Active and passive transport. Phloem loading involves active transport of photoassimilates from source tissues to phloem with the help of proton pumps, ATPases, and other transport proteins. However, passive diffusion may also play a role in some cases. Then, regulation by hormones and environmental factors. Phloem loading is regulated by plant hormones such as cytokines, auxins, and abscisic acid, as well as environmental factors such as light and temperature. And then different loading mechanisms. Phloem loading occurs via different mechanisms in different plant species. For example, in some species, photoassimilates are loaded directly into the sieve tube elements, while in others they are first loaded to companion cells before being transferred to sieve tube elements. Examples of phloem loading can be seen in many plants such as sugar beet, where sucrose is loaded into the phloem from source leaves and transported to the trap roots for storage. In addition, phloem loading is important for the growth and development of fruits and seeds in many plant species. The importance of phloem loading lies in its role in regulating the distribution of photoassimilates within a plant. By ensuring that source and sink organs are appropriately connected, plants are able to optimize their growth and development as well as respond to changing environmental conditions. Node gene. Node genes are a group of genes that are present in rhizobial bacteria and play a crucial role in the formation of fruit nodules in leguminous plants. These genes encode proteins involved in the biosynthesis of node factors, which are signal molecules that induce the formation of root nodules. When a leguminous plant such as a soybean or clover is exposed to rhizobial bacteria in the soil, the bacteria produce node factors which are perceived by the plant's root hairs. The perception of node factors triggers a cascade of events that led to the formation of root nodules. Inside these nodules, the rhizobial bacteria convert atmospheric nitrogen into a form that is usable by the plant, a process called nitrogen fixation. In node genes, the node genes are organized in a cluster on the bacterial chromosome, and their expression is regulated by a variety of environmental and physiological signals. The expression of these genes is induced by the presence of flavonoids in the plant root exudates, which act as signal molecules that activate the transcription of the node genes. The node genes encode a range of proteins involved in the biosynthesis of node factors, including enzymes that modify the lipid and carbohydrate moieties of the node factors. The node factors produced by different rhizobial species are highly diverse and can vary in the length and complexity of their carbohydrate chains, as well as in the nature of their lipid moieties. This diversity is thought to play a role in the specificity of the symbiotic interaction between the rhizobia and the leguminous plant. Overall, the node genes and the node factors that produce are essential for the establishment of the symbiotic relationship between rhizobia and leguminous plants. This relationship allows the plant to access a new source of nitrogen, which can increase growth and yield while reducing the need for synthetic fertilizers, which can have negative environmental impacts. The node genes are a group of genes found in rhizobia, a type of bacteria that form mutualistic relationships with certain plants, particularly legumes. These genes play a critical role in the formation of root nodule, specialized structures that allow for the exchange of nutrients between plant and the bacteria. The node genes are involved in a process known as nodulation, which begins when the rhizobia are attracted to the plant root by chemical signals released by the plant. Once the bacteria have colonized the root surface, they produce node factors, which are specific signaling molecules that stimulate the plant to begin the process of nodule formation. The node genes are responsible for the synthesis of various enzymes and other proteins involved in nodulation. These include enzymes that synthesize node factors, as well as proteins involved in the transport of node factors from the bacteria to the plant, and the synthesis of other compounds that promote nodule formation. The importance of node genes lies in the fact that they allow for the formation of a mutualistic relationship between plant and the rhizobia. The bacteria provide the plant with nitrogen, which is essential for growth and development, while the plant provides the bacteria with a source of energy in the form of carbohydrates. Examples of legumes that form nodules with rhizobia include soybeans, clover, and peas. The ability of these plants to form nodules and fix nitrogen is of great agricultural importance, as it reduces the need for fertilizers and promotes sustainable agriculture. Nitrogen fixing organisms in detail. Nitrogen fixing organisms are organisms that convert atmospheric nitrogen into a form that can be utilized by plants. This process is known as nitrogen fixation, and it is important because nitrogen is an essential nutrient required for plant growth and development. There are two main types of nitrogen fixing organisms, like symbiotic and non-symbiotic. Symbiotic nitrogen fixation. Symbiotic nitrogen fixation occurs when nitrogen fixing bacteria form a symbiotic relationship with leguminous plants. This relationship is mutually beneficial, as the bacteria provide the plant with a source of nitrogen and the plant provides the bacteria with a source of energy in the form of carbohydrates. The process of symbiotic nitrogen fixation can be broken down into the following steps, like recognition and attachment. The bacteria recognize and attach to the root hairs of the plant. Infection. The bacteria penetrate the root hair and enter the root cortex, where they form nodules. Then nodule formation. The nodules are formed as a result of cell division in the root cortex. Then nitrogen fixation. The bacteria convert atmospheric nitrogen into a form that can be utilized by the plant. Then nitrogen assimilation. The plant incorporates the fixed nitrogen into its metabolism. Examples of symbiotic nitrogen fixing bacteria include rhizobium, bradyrhizobium, and xenorhizobium. Bradyrhizobium, xenorhizobium, and rhizobium. Non-symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation occurs when a free-living bacteria fix atmospheric nitrogen without forming a symbiotic relationship with the plant. The process of non-symbiotic nitrogen fixation can be broken down into the following steps, like nitrogen fixation. The bacteria convert atmospheric nitrogen into a form that can be utilized by plants. Then nitrogen assimilation. The fixed nitrogen is incorporated into the metabolism of bacteria or surrounding plants. Examples of non-symbiotic nitrogen-fixing bacteria include Azotobacter and Cyanobacteria. Overall, nitrogen fixation is an important process for plant growth and development, and nitrogen-fixing organisms play a critical role in maintaining nitrogen levels in the soil. Then, Agrobacterium, EGH in detail. So, Agrobacterium is a genus of gram-negative bacteria that is widely used in biotechnology for its ability to transfer DNA between organisms. It is known to cause crown gall disease in plants, which results in the formation of tumors on the stems and roots of the infected plants. Next, agrobacterium mediated transformation is a genetic engineering technique used to introduce foreign genes into the plants. It involves the transfer of a DNA fragment from a plasmid of Agrobacterium tumefaciens to the plant genome. The transferred DNA fragment contains the desired gene of interest as well as other genomic genetic elements necessary for integration and expression of the gene in the plant. Natural transformation. Agrobacterium can naturally transfer DNA to plants, causing them to develop tumors. The process occurs through the use of a specific genetic system known as TI, tumor inducing plasmid. Then, genetic engineering tool. Agrobacterium is widely used in genetic engineering as a tool for the transfer of DNA to plants. This is because it can effectively deliver the DNA to the plant cells and ensure that the transferred genes are integrated into the plant genome. Then tDNA, the DNA fragment that is transferred from Agrobacterium to the plant genome is known as tDNA or transfer DNA. It contains the desired gene of interest as well as other genetic elements that allow the gene to be expressed in the plant. Then agroinfection. The process of introducing foreign DNA into a plant using Agrobacterium is known as agroinfection. It involves the use of a specially prepared Agrobacterium strain that has been modified to contain the desired gene of interest. Then importance. Agrobacterium mediated transformation is a valuable tool for plant breeding, genetic research and biotechnology. It has been used to produce crops that are resistant to pests and diseases as well as crops that have improved nutritional value or other desirable traits. An example of the use of agrobacterium in genetic engineering is the production of insect-resistant cotton. The cotton plants are transformed with a gene from the bacterium Bacillus thuringiensis, which produces a protein that is toxic to certain insect pests. The gene is introduced into the cotton plant using agrobacterium-mediated transformation, resulting in the production of cotton plants that are resistant to insect damage. Flower development. Flower development refers to the process by which a flowering plant produces its reproductive structures, including flowers, fruits, and seeds. It involves complex molecular and genetic interactions that result in the formation of specialized organs that facilitate sexual reproduction. So, initial stage. In this stage, cells in the meristem region of the plant start to differentiate into floral meristems. The floral meristem is a specialized region that is responsible for forming flowers. The initiation stage is often triggered by environmental and developmental cues such as changes in temperature, light, or photoperiod. Then patterning stage. During this stage, the floral meristem develops into distinct whorls, each of which will eventually give rise to different floral organs such as sepals, petals, stamens, and carpels. The patterning of floral organs is regulated by a complex network of genes that interact with each other to form specialized floral structures. Differentiation stage. In this stage, cells within each floral organ begin to differentiate into specialized cell types such as epidermal cells, parenchyma cells, and vascular cells. The differentiation process is tightly regulated by various hormones such as auxins, cytokines, and gibberellins. Then growth stage. During this stage, floral organs continue to grow and mature, and their final size and shape are determined. Hormones such as gibberellins, auxins, and abscisic acid play important role in regulating floral growth. Then reproductive stage. This is the final stage of flower development in which a flower is fully matured and ready for reproduction. The reproductive structures, including the stamen and carpel, are fully formed and the flower is ready for pollination. Once pollinated, the ovary develops into a fruit and the ovules inside of the ovary develop into seeds. Next important points. Flower development is a complex process that is tightly regulated by a network of genes and hormones. Environmental and developmental cues such as light, temperature, and photoperiod play important roles in initiating flower development. The patterning of floral organs is regulated by a complex network of genes. And hormones such as auxins, cytokines, gibberellins, and abscisic acid play important roles in regulating floral development. Once fully matured, flowers are ready for pollination and reproduction. And examples, floral development varies widely among different plant species, but the process generally follows a same basic stage. In Arabidopsis thaliana, a small flowering plant commonly used as a model organism in plant research, flower development involves the sequential formation of sepals, petals, stamens, and carpels. In maize, flower development is more complex, and flower development and maize include with separate male and female inflorescence developing on different parts of the plant. Flower development is crucial for the development and survival of flowering plants. By producing flowers, plants are able to attract pollinators and ensure the fertilization of their seeds, which is necessary for the production of future generations. In addition, flowers are an important source of food and shelter for many animals, making them an integral part of many ecosystems. Understanding the molecular and genetic mechanisms that underlie flowering development is therefore critical for improving crop yield, conserving endangered plant species, and maintaining healthy ecosystems. Thank you for listening.